Brian Reckenbeil from Florida Fish and Wildlife Conservation Commission.
All right, thank you, Megan, for the introduction. And I first want to acknowledge my two co-authors, Colette Feehan, who is a collaborator from Rutgers University, which is located up in New Jersey, as well as Bill Sharp. And we work for the FWC down in Marathon in the Middle Keys, and we are part of the Restoration Ecology team. As Elliot talked about before, he worked on sponges. We work on a wide array of topics. And what I'm talking about today specifically in relation to coral reef restoration is some of the ecological processes that we work with. Thus far, as Tom kind of mentioned, the primary focus of coral reef restoration has been on restoring corals. And the coral reef community has been really successful in the coral husbandry, coral nursery operations. And in Florida, a large network of partners from Key Biscayne all the way down to the Tortugas have outplanted numerous amounts of corals. However, it has become increasingly recognized that restoring corals alone may not really be restoring ecological function to these reefs. So researchers, many of the scientists here in this room, have been starting to incorporate ecological processes into their work. And we at FWC, in our Restoration Ecology Lab, have been working on two of these ecological processes. And the two I'm talking about today are herbivory and predator-prey dynamics. And the first one I'm going to talk about is herbivory. When we think of herbivory in the Caribbean region, what comes to mind is the animal Diadema antillarum. And as we all know, they had a population crash in the early 1980s, which has exacerbated the amount of algae growing on the reefs, which has helped exacerbate coral declines. And back in 2009, Martin Skip Moe, in affiliation with Moat Marine Lab, began to culture Diadema in his laboratory. And we got really excited when we found out that he was able to successfully make a cohort grow out to an early benthic juvenile stage. And see in some of these photos, some very small Diadema he was able to culture. And so the first thing we kind of came to mind was, are we now going to be able to use these juvenile Diademas produced in a hatchery setting in a more comprehensive reef restoration strategy? And when you think of animals that are produced in a hatchery setting, you kind of need to think about if they are ecologically and phenotypically the same as their wild counterparts. And often that's not the case. And because when you're working with hatchery reared individuals, our agency, FWC, requires there be some best management practices. And these include both a health protocol for your species you're working with, as well as you need to evaluate the population structure. So some work that we have done so far in the laboratory is we've done a little bit of experimental work, and we have noticed that there are some hatchery-induced deficits. However, we think that via conditioning and different grow-out habitats, we can overcome that. Here's two photos. You can see this is a wild urchin, lots of spine density. Hatchery reared individual has a lower spine density. And thinking about protection at the reef, an individual probably with more spines might be able to defend itself a little bit better. 2 years ago, in 2015, FWC completed a health manual, so we're all set to go from a health standpoint, as well as in the last 2 or 3 years, we surveyed Diadema from the Tortugas up to Key Biscayne and found out that there is, in fact, one genetic population. So what that means for the future is that we do not have to worry about, if we are able to hatchery rear some individuals, we can kind of put them anywhere we want because it's one genetic population. However, where we are today, after initial success that Martin Moe had, this husbandry process has stalled. There's only been a few cohorts of Diadema that have been able to successfully grow out to this early benthic stage. However, we have a great partner, the Florida Aquarium, that is continuing this effort in the terms of trying to overcome whatever bottleneck is preventing Diadema from reaching this early benthic stage. Another problem that is kind of well known in the literature is that throughout the Caribbean region, reefs are degrading and declining in structure. This is one of our research sites, Delta Shoal. As you can see, it's kind of flat. There's not much rugosity left here. So when we think of Diadema in the future, we have a loss of reef architecture, and that may help are maybe limiting some of their habitat that they need to survive. So with that in mind, in 2015, we began partnering with Rutgers and Colette Feehan, and what we have been doing here has been looking at larval influx dynamics. And us at FWC, we've been monitoring 2 sites for a year and a half. We've been setting up these little turf collectors, and here's Colette removing a collector, putting it in a bag, and bringing it back. And we've been monitoring these sites every month. So we go there every month, take the turfs off, replace it with new ones, and bring them back into the lab. And our 2 sites are Delta Shoal, I just pictured before, and another inshore site. And what we have found at our 2 sites, at Delta Shoal, represented by the black line, you can see we have a higher influx of larvae in the water than we are finding at our second site. So one site we're calling more of a high larval flux inflow juvenile diadema on the turf plates versus our second site. Now what's interesting when we go out to these sites and do a benthic survey, this blue bar over here on the right, that's our Delta Shoal, so that is where we had a high influx of larvae. We find 0 juvenile or 0 diadema on the benthic environment when we survey the bottom. However, when we look at our site at Washerman, this red site here, we are finding lots of individuals. And this is a site where we have lots of habitat that we think provides shelter for these diadema. And then we have another comparative site a few hundred meters away that has low habitat environment, and we're not finding many individuals on the bottom. So it's kind of interesting to see. This again might show that we're habitat limited in terms of being able to grow out diadema. So being that we might be habitat limited, where does this bring us in the future in terms of coral reef restoration? So if we're going to be continuing to outplant corals and want diadema in those areas, we need to consider like sheltering habitats for these diadema animals. So we did some pilot scale work a few years ago. This is a conceptual design of our research, one of our research trials. We took some diadema, we tethered them to the bottom with some monofilament and a nail so they couldn't escape. And half of those animals we gave shelter to. So we took a clay pot, we cut it in half, we put it on the bottom, made sure it didn't move, we strapped that down. And you can see this is again Delta Shoal, here's a pot, here's a pot, here's a pot. And as I said, half of them had shelter. As you might expect, animals with shelter, represented by the orange bar, survived a lot longer than the individuals that had no shelter. Again, kind of showing that shelter does help in the longevity of persistence of diadema. So our next steps for us at FWC for when we are going to continue to outplant our Acropora cervicornis, we're also going to be placing clay pots out there and potentially diadema in these restoration sites so that we can have more of a holistic dynamic, hoping that the diadema will eat the algae and that won't compete as much with the corals. So now I'm going to transition to the second part of my talk, and that is on the ecological processes of predator-prey dynamics. And as we all know, those of us who continue to monitor coral colonies, snail predation is a big problem throughout the region. And we find Coralophilia abbreviata preying readily upon our corals. And it really is impractical from a monitoring standpoint and longevity of our outplants to go out there and pick corals off continually for years at a time because nobody really has the money to monitor every outplant forever. So because of that, a few years ago, Bill actually noticed on some of our colonies, C. abbreviata was being preyed upon by a gastropod called Thais deltoidea, this animal here. And so he thought, oh, this is great. Now something might be able to eat these snails that are preying upon the coral. So we brought a bunch of them back into the laboratory, and we're thinking, can we now use this as a tool to increase restoration success in the future if we can use these Thais? So the next question that came to mind is, does the presence of Thais reduce predation by C. abbrev on Acropora cervicornis? So we went to our coral nursery. We brought in some coral fragments that had broken off of trees. They were just laying there in the sand. We might not know what genus they were, and brought them into the laboratory to test this experiment. And what we found, lo and behold, the Thais prevent the C. abrevia from being on the coral. So when there's no Thais, the predator coral snail spends a lot of time, about 70% of its time over a month they spend in the lab on coral, versus when there was 1 or 2 predators in the tank, the amount of time they spent on coral was significantly less. When we look on, this is a percent of live coral tissue by the end of the month-long trial. Again, when there's 0 Thais in the tank, almost all of the coral was eaten. When you have 1 or 2 predators in the tank, upwards of 80–90% of the coral remained alive after 1 month. So it's really promising results that we saw in the laboratory. So our next step is, this is all good and done in the lab, but what are we going to see out in the field? So we went to a site off of Key Largo at Davis Reef, and we outplanted 20 acropora colonies. And each of these colonies we stocked with 5 Corallophilia abbreviata. And then again, half of these colonies, represented by the red dot, was stocked with 2 Thais, and that's the predator snail. And what did we find? We found same results. When there was Thais absent, the corallivores spent more time on the coral, 75% of the time. In the plots that had Thais present, under 50%, maybe 40% of the corals were actually on, or the snails were actually on coral. Again, when this relates back to the percent of live coral tissue, when Thais are absent, you have less survival because the snails go and prey upon the coral. When the Thais is present, more of the coral remains alive. And this study was performed for 2 weeks out in the field. So when we think of continuing our coral restoration work, we think of ecological processes and how they may be beneficial for the future, because as Tom has mentioned, just strictly putting out numbers and numbers of corals may not really be long-term sustainable, but including some of these dynamics may be helpful for the future. On the lungs of herbivory and diadema, we still have a long way to go, but we're hoping Florida Aquarium is going to break through that bottleneck to begin to successfully culture diadema more and more. And then as well as we need to evaluate our study sites. Is there enough shelter for diadema at a study site? If you don't have shelter, kind of see these flat areas, they get picked off pretty quickly. We're thinking, snail predation, using predatory snails, finding sites with snails, these Thais sites. That might be an untapped resource for helping control predation. And with that, thank you, and I guess you don't have any questions at the panel.